Welcome to Educon. My name is Imam, and in this session we'll discuss about the M phase, that is mitosis phase, which is most important phase where exactly the cell division occurs in cell cycle. So coming to this session, this is the M phase is the most dramatic period of the cell cycle, involving major reorganization of virtually all components of the cell. So here, this is important point here. Okay. So this is the most dramatic period of the cell, involving major reorganization. Reorganization means arranging all the different cellular organelles, and also the division of nucleus, everything occurs in this phase. Since the number of chromosomes in the parent and progeny cells is the same, it is also called equational division. Okay, remember this equational division. Okay. Though for convenience, mitosis has been divided into 4 stages of nuclear division. That is, we call, what is a nuclear division is called, the other term for nuclear division is karyokinesis. Okay. It is very essential to understand that cell division is a progressive process, and very clear-cut lines cannot be drawn between various stages. So there is no perfect, start and end of every stage. So we can't draw a particular lines. So this is this stage, this is that stage, like that. So it is very progressive process, and very clear cut lines cannot be drawn between various stages of M phase. The karyokinesis involves 4 stages: prophase, metaphase, anaphase, and telophase. So all these are the 4 phases. How many phases are there in M phase? 4 phases. That is, how many stages are there in karyokinesis? Same. So the another name for this for nuclear division, division is karyokinesis. Coming to prophase, which is the first stage of karyokinesis. This is the starting line, is very important. Yeah, what is the first stage of karyokinesis? It is the prophase. So prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phases of interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but intervene. Okay, and prophase is marked by the initiation of condensation of chromosomal material. What is this line here? Okay, condensation of chromosomal material is, what is this term called, is reorganization of, for example, reorganization of chromatin into chromosomes. So condensation of chromosome materials. At the time, what is there? There is chromatin before. Okay, it is proteinaceous, strands. So this chromatin will turn into chromosomes. That is what called. So prophase is marked by the insertion of condensation of chromosomal material, means reorganization of chromatin into short chromosomes. Okay. The chromosomal material becomes entangled during the process of chromatin condensation. Okay, before how will be the chromatin present in the nucleus during the interphase? It will be like this. Okay, slowly it will be organized. This is called chromosomal condensation. Okay, now understand. Okay. The chromosomal material become entangled. Entangled means released during the process of chromatin condensation. The centrosome, which had undergone duplication during S phase, okay, of interphase. Okay, remember this line. The centrosome, which had undergone duplication. Where exactly the centrosome duplicate itself? That is S phase of centrosome duplication occurs in S phase of interphase. S phase of interphase. Okay, this is an important point. Okay, now the centrosome which had undergone duplication during S phase of interphase now begins to move towards the opposite poles of the cell. So when does the centrosome start moving towards the opposite poles of the cell? In which stage? In prophase. Okay, in prophase of meiosis. So in M phase we have totally 4 phases. So in prophase it starts moving towards the opposite poles of the cell. The completion of prophase can thus be marked by the following characteristic events. So the completion, how we'll define the prophase is there or it is finished, it is completed. Here these are the important events which occur in prophase. Chromosomal material condenses to form compact mitotic chromosomes. Okay, now understand, before I told you condensation of chromosomal material is from chromatin like this to then like this. Okay, it occurs in prophase. Chromosomes are seen to be composed of 2 chromatids attached together at centromere. So this is also important point. Okay, chromosomes are seen here, the centromere is in the center. Okay, it holds 2 chromatids together. Okay, centrosome which had undergone duplication during interphase, okay, begins to move towards the opposite poles of the cell. Each centrosome radiates out microtubules called asters. Okay, this term is also very important. We need to remember this. Okay, here around 4 to 5 lines are so much important, very important. So they may come in a question form. The 2 asters together with spindle fibers forms mitotic apparatus. So in prophase, 2 major characteristic of events occur in prophase, that is the condensation of chromosomal material and the moving of centrosome towards the opposite poles of the cell. So here we need to remember centrosome duplication occurs in S phase of the interphase. Remember, don't don't get confused here. Okay. Okay. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complex. Endoplasmic reticulum, nucleolus, and the nuclear envelope. Okay, cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. There is no any membrane. So this is also we have around 4 to 5 important lines from this section. Okay, 10.2.1 from prophase. Coming to metaphase. So here in the image, in the picture, you can see how the prophase is there. Okay, here these are the chromatin. Okay, and then organized into chromosomes. Okay, the complete now coming to metaphase, the complete disintegration of nuclear envelope marks the start of second phase of mitosis. So complete disintegration of the nuclear envelope, which marks the start of the second phase of the mitosis. Okay, hence the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. In metaphase, in prophase, we see condensation of chromosomes occurs. Here it is finished. By this stage, you can see that under the microscope. This is the stage at which morphology of chromosome is most easily studied. Okay, now at which stage the chromatin can be studied, better way. This is the metaphase. Okay, not prophase, not other phase. This may also come as a question. Okay, sometimes the questions will be so simple in NEET. You may appear it, but unfortunately, the options will be so similar. So you'll get confused. So we need to remember these are the basic points to remember when you are appearing for NEET. So in this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. At this stage, the morphology of chromosome is most easily studied. Okay, morphology of chromosome is most easily studied. At this stage also, metaphase chromosome is made up of two sister chromatids which are held together by the centromere. Okay, and you can see in the image. Okay, slowly we can see all the images. Small disc-shaped structures at the surface of the centromeres are called kinetochores. Okay, we studied about the kinetochores and centromere in the first unit also in nucleus, while we discuss about the nucleus. These structures serve as the site of attachment of spindle fibers. So at these junctions, what happened? The spindle fibers, they will come and they get attached. Okay, the spindle fibers to the chromosome that are moved into the position at the center of the cell. Okay, now you understand. So this is the centromere. Okay, and these are the spindle fibers, and they are holding the chromatids. Okay, now see, these structures serve as a site of attachment of spindle fibers formed by the centromere to the chromosome that are moved into the position at the center of the cell. Now what happened? They hold together and it appears to be in the center. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator. So, at which state the chromosome lie at the equator? That is in metaphase. Okay, in M phase, prophase we have, metaphase we have, other two stages we have, but there is one stage at where the chromosomes will come and lie on the center of the cell. That is a lie at the equator with one chromatid of each chromosome connected by its kinetochore to the spindle fibers from one pole to its sister and its sister chromatid connected by its kinetochores to spindle fibers from opposite pole. So here let's read the sentence again. And the metaphase is characterized by all the chromosomes to lie at the equator with one chromatid of each chromosome connected by its kinetochore to a spindle fibers from one pole, and its sister chromatid connected by its kinetochore to spindle fibers from opposite pole. For example, now you see here, this is the two chromatids. So what happened? We have a centromere in this position, centromere in this position. So spindle fibers, they will come and attach here. So same like this, from the sister chromatid also attached on this side. Understand that? This is the meaning of that sentence. The plane of alignment of chromosome at metaphase is referred to as the metaphase plane. Okay, the plane of alignment of the chromosomes at the metaphase is referred to as metaphase plane. The key features of metaphase are spindle fibers attached to the kinetochores of the chromosome. Okay, this is the most important lines. Okay, chromosomes are moved to spindle equators and get aligned along the metaphase plate through the spindle fibers of the both poles. Okay, now you can see the chromosomes are arranged at the center, the spindle fiber arising from the centromere, they hold it and it forms a metaphase plate. Okay, anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as daughter chromosomes of the feature of the daughter nucleus, begin their migration towards the two opposite poles. That we'll see in the next slide. Okay, as each chromosome moves away from the equatorial plate. So when in what phase the chromosomes move from the center to the other side, that is the towards the opposite poles. In metaphase, what we see all the chromosomes lie at the equator of the cell, whereas in anaphase it starts moving from the equator. Okay, this we must remember. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge. So for example, if something is pulling like this, so what, for example, it is the chromatid. If we are pulling, what happened? This will bend like this. Okay, it will be like this position when you are pulling it. Okay, with the arms of chromosome trailing behind. Yes, when you pull this, these are the arms of chromatid. It will be trailing behind. Thus, anaphase stage is characterized by the following key events. What are those events? Centromere splits and chromatids separate. Okay, so chromatids move to opposite poles. Now you can see here how clearly it is. Now we understand what exactly occurs in anaphase. Okay, coming to telophase. At the beginning of the final stage of karyokinesis, that is telophase. What is the final stage of the karyokinesis? Is the telophase. The chromosomes that have reached their respective poles decondense and lose their individuality. Okay, the individual chromosome can no longer be seen, and each set of chromatin materials tends to collect at each of the two poles. Okay, this is a stage which shows the following key events. So here what happened? Before we see condensation of chromosome. Okay, from chromatin to chromosome occurs. Again here in this stage, the chromosomes that have reached their respective poles decondense and lose their individuality. Okay, the individual chromosomes can no longer be seen, so we cannot see individual chromosome in telophase, and each set of chromatin materials tends to collect at each of the two poles. So what happened? They are tied up with the centromere and kinetochore, so they will lose. Here what happened? The chromosomes, they decondense. Okay, and here this is how after decondensation it will be like this. So there is no any specific chromosome we can observe. Okay, so the individual chromosome can no longer be seen, and each set of chromatin materials which is present on this the one side and other side, okay, tends to collect at each of the two poles. This is a stage which shows the following important events or key events. What are those key events? Chromosome cluster at opposite spindle poles, and the identity is lost as a discrete element. So there is no any more chromosome here. Again, we will come back to chromatin. Nuclear envelope develops around the chromosome cluster at each pole, forming two daughter nuclei. Okay, then we have other organs such as nucleolus, Golgi complex, and endoplasmic reticulum. They reform again. With this, we have concluded this M phase. So key important points we need to remember from these sections are in NCERT biology. Okay, when you are reading NCERT, please read it carefully and slowly, and after going through a video lecture, you just read the book properly and try to understand the concept. Okay, so in prophase what happened, in metaphase, in anaphase and telophase, what exactly is happening in these phases. Okay, and thank you, and please subscribe to my channel, and which will help me to bring daily updates on NEET and also your biology classes. Thank you.